Good morning, ma'am. My name is Kartik Rajput. My roll number is 013, group 71. For today's presentation, my topic is photorespiration. Photorespiration occurs when carbon dioxide level inside the leaf becomes low. This happens on hot dry days. On hot dry days, the plant loses excess water, and to prevent these, it closes its stomata to prevent the water loss. The plant continues to fix carbon dioxide with its stomata are closed. The carbon dioxide will get used up, and the ratio or oxygen ratio in the leaf will increase relative to the carbon dioxide concentrations. When carbon dioxide level inside the leaf drops to around 50 ppm, Rubisco starts to combine oxygen with RuBP instead of carbon dioxide. The net result of this is that instead of producing 2 to 3 PGA molecules, only one molecule of PGA is produced and a toxic 2C molecule called phosphoglycolate produced. So this diagram shows the Rubisco reaction when there is less oxygen in the leaves. Next is what is phosphoglycolate. The plant must get rid of the phosphoglycolate since it is highly toxic. It converts the molecule of glycolic acid. The glycolic acid is then transported to the peroxisome and then converted to glycine. So this diagram represents the phosphoglycolate conversion to serine. The serine is then used to make other organic molecules. All these conversions cost the plant energy and results in the net loss of carbon dioxide from the plant. To prevent this process, two specialized biochemical additions have been evolved in plant world called C4 and CAM metabolism. So the C4 pathway is designed to efficiently fix carbon dioxide at low concentrations, and plants that use this pathway are known as C4 plants. These plants fix carbon dioxide into 4 carbon compounds C4 called oxaloacetate. This occurs in cells called mesophyll cells. A CM plant lives in very dry conditions and, unlike other plants, open their stomata to fix carbon dioxide only at night. Like C4 plants, they use PEP carboxylase to fix carbon dioxide forming oxaloacetate. The oxaloacetate is converted to malate, which is stored in cell vacuoles. During the day, when the stomata are closed, carbon dioxide is removed from the stored malate and enters the Calvin cycle. This is a diagram. This figure shows the stomata difference at night and day. So the difference between Kelvin, C3 and C4. C3 temperature, the temperature lies between 15 to 25 degrees Celsius. In C4, the temperature lies between 30 to 35 degrees Celsius. There is absence of malate in C3, whereas the malate is present in C4. One carboxylation reaction, whereas in C4, 2 carboxylation reactions occur. At CO3 is the substrate in C4, whereas in C3, CO2 is the substrate. The closed stomata, C4 closed stomata to reduce the water loss and concentrating carbon dioxide in the bundle sheet cells. In C3, the leaf structure is normal and same as in other leaves. Additional ATP is required in C4. So this is the C4 and CAM plants difference. Next is comparison between C3, C4 and CAM. In C3, G3P day and night, malate in C4 and malate in as well as malate in CAM also. Anatomy: no bundle sheet cells in C3. C4 has the bundle sheet cells and CM no bundle sheet cell. Number of stomata in C3 are 2000 to 31,000. In C4, 10,000 to 16,000, and in CAM 100 to 800. Photorespiration up to 40% in C3, not detectable in C4 and not detectable in CM. Species wheat, rice, potato. In C4 contains sugarcane, and CM pineapple, vanilla, cactus. Factors affecting photorespiration: oxygen to carbon dioxide ratio. If cells have low oxygen but higher carbon dioxide, normal photosynthesis, that is Calvin cycle, dominates. C4 plants have little photorespiration because they carry the carbon dioxide to the bundle sheet cells and build up high carbon dioxide. Calvin cycle reaction always favored over photorespiration. If cells have higher oxygen and lower carbon dioxide, photorespiration dominates. Temperature. Photorespiration increases with temperature. Next is regulation of carbon dioxide fixation. Plant cells have chloroplasts that carry out photosynthesis that converts carbon dioxide to glucose. Plant cells also have mitochondria that carry out glycolysis, TCA, and oxidative phosphorylation that converts glucose to CO2. To prevent futile cycling of carbohydrate, cells must regulate the activities of key Calvin cycle enzymes. These enzymes respond indirectly to light activation. Light energy is available, then the Calvin cycle proceeds. If no light energy is available, no fixation of CO2 occur. Among the key changes that regulate Calvin cycle versus respiration are environmental factors like light, intensity, temperature, and availability of hydrogen and carbon dioxide and oxygen. Cellular factors that affect it are cell state of key metabolism, NADPH, ATP inhibitors, reducing power, etc. That's all for my PPT. Thank you.